Welcome to Chapter 4. This chapter is really all about cells. So we're going to talk about cell theory, we'll talk about cell size and scale, the microscope, which is how we see cells, prokaryotic versus eukaryotic cells, and different cell organelles. Our book really doesn't talk much about cell theory, but it's pretty important. It's one of the key theories of biology. So cell theory says that all organisms are made of at least one cell. It also says cells are the simplest collection of matter that can be considered alive, and all cells come from pre-existing cells. Now, that doesn't explain how we first got cells, but it does talk about how we continue to have cells. So even though cells can be considerably different, they do share several common features, which we'll get talking about coming up here. So with the size of cells, really the metabolic requirements, so the requirements of the various chemical reactions and their waste products and the nutrients that they need in the cell really kind of sets the upper limit on cell size. So small cells have more surface area relative to their volume. You can kind of see here in this picture, right, we've got several small cells that make up the same volume as all these small cells make up the same volume as this larger cell. These smaller cells have more surface area. So that's more surface area for nutrients to go into the cell and waste to leave the cell. Most cells are actually too small to be seen without the use of a microscope. So this link at the bottom of the page does a good job kind of showing you cell size and scale, and you can kind of play around with it a little bit. But really, when we talk about cells, right, cells are very small. We've talked about lots of very small things so far because we've talked about atoms and molecules. But if we look at cells, they're much larger than atoms and molecules because cells are made up of, many atoms and molecules, although realistically, we would all probably consider a cell to be pretty small. So if we look at this size and scale thing here, right, like there's a coffee bean, that's small. There's a grain of rice, that's small. A sesame seed, that's small. But really on the scale of cell type things, they're pretty large. So if we zoom in, here's a grain of salt. Here's an amoeba, which is a pretty large single-celled organism. If we go farther, here's the paramecium. Here's the human egg. Again, all pretty large cells. Human skin cell, photoreceptor, like if you've heard of rods and cones in your eyes, this is a rod. If we keep zooming in here, here's a red blood cell. Here is baker's yeast, so the yeast that makes your bread rise. Here's the old powerhouse of the cell, right, the mitochondria. Here's E. coli, which is a bacterium. And then we've got measles, which is a virus, HIV, influenza, all viruses, phage, which is a virus that infects bacteria, right? So viruses are smaller than cells. And then if we zoom in, a ribosome is an organelle that we'll talk about coming up, and that's, smaller than a cell. Then we've got antibodies, which is a protein, tRNA, which is a type of RNA, hemoglobin, a protein, a phospholipid, which makes up the cell membrane, and kind of so on and so forth here all the way till we get to the carbon atom. So I don't want you to memorize the exact size and picometers or whatever of these different structures, but I do want you to have a scale in your mind of all of these small things, which are smaller and which are larger. In order to see cells, we need microscopes. There are certain parameters of microscopy. The magnification that we have is the ratio of the object's image size to its real size. I think people are pretty aware of resolution now. Resolution is the measure of clarity of the image or the minimum distance between two distinguishable points. And then contrast is the visible differences in the parts of a sample. So there's two kinds of microscopes, broadly speaking. We've got light microscopes and electron microscopes. Light microscopes use light. They pass that light through a specimen through glass lenses, and they can magnify effectively to 1,000 times total magnification, but many subcellular structures are really too small to be viewed with a light microscope. Then there's electron microscopes, which instead of using light, they use focused beams of electrons. And then with electron microscopes, there's scanning electron microscopes, which view the surface of a specimen and kind of look three-dimensional with the images. And then there's transmission electron microscopes, which view internal structures of cells. So let's look at these microscopes and these different images of E. coli and see which kind of microscope could produce which kind of image. So the first microscope up here, this is kind of like the microscope you would have in our lab, and this is a light microscope. So which of these three images looks like it could be produced with this kind of microscope? So that's going to be this image here, or C. So that's what E. coli looks like under a light microscope. That means this other microscope is going to be an electron microscope. So that goes for both of these two here. And then we've got different kinds of electron microscopes, right? There's transmission electron microscopes and scanning electron microscopes. So with the transmission electron microscope, that shows us kind of internal structures. So B is going to be from a transmission electron microscope. So that means A is going to show us those kind of superficial surface features. So that's going to be from a scanning electron microscope there. We've already talked about prokaryotic and eukaryotic cells, but here's a little bit more information on prokaryotic versus eukaryotic cells. Remember again that root word pro means before. The root word karyotic is referring to the nucleus, and the root word eu means true, which is again that's terrible handwriting. Eukaryotic, true, karyotic nucleus. So these cells have a nucleus. Prokaryotic cells do not have a nucleus. Prokaryotic cells are largely more simple than eukaryotic cells. They don't have membrane-bound organelles. Eukaryotic cells do have membrane-bound organelles. Prokaryotic cells are smaller. Eukaryotic cells are larger. Examples of prokaryotic organisms include bacteria and archaea, and eukaryotic cells include animals, plants, protists, and I don't know why I didn't list it, but fungi as well are eukaryotic organisms. And if we look at this image down below here, it's showing the prokaryotic cell with the single circular chromosome and no nucleus. Eukaryotic cell has that nucleus. But I think it's worth mentioning that if we look at the scale bars on both of these images, they're not set to the same scale. So it's showing the prokaryotic cell is smaller, but in reality, they're actually much, much smaller. This image does a better job showing you how much smaller the prokaryotic cell is compared to the eukaryotic cell. And then up here, we have a real image. These big cells that kind of look like sort of like a lava lamp, those are eukaryotic white blood cells, and these smaller little specks, the darker red specks here, those are prokaryotic cells. So which of these cells is prokaryotic and which of these cells is eukaryotic? These first cells on the left here, these are going to be eukaryotic. They have a nucleus and they're much larger. These cells on the right, you can't see the nucleus. You know, if you take a microbiology class or even in Bio 2, you'll learn that there's sort of certain shapes that prokaryotic cells usually are in. They're in kind of this rod shape often, or this circular shape. So these are pretty common prokaryotic cell shapes. So on the left we had eukaryotic, and then on the right we had prokaryotic cells. So what about this picture? If you want, you can pause it. So on the left, prokaryotic. On the right, eukaryotic. You can't necessarily tell maybe from the picture like which one of these intercellular structures here are the nucleus, but since you can see so many of them, that gives you a good hint that that is eukaryotic. So the next part of our lecture, we're going to talk about cellular structures like cell organelles, and this is a course objective. So this will definitely be on the test. These make good matching questions and good kind of situation type questions. So the course objective is to describe basic cellular structures and their functions. So for all of these cellular structures, I want you to focus on the structure, like what it's physically made out of, the location where you can find it, and the function. I don't want simile or metaphor type answers, and I won't have simile or metaphor type examples in a matching question. What I mean by a simile or metaphor type question or answer is when people say, oh, the nucleus is like the brain, or the mitochondria is the powerhouse of the cell. Those might be helpful when you're memorizing these structures, but I want you to know what they're actually made of and what they actually do. I'm going to break things down a little bit differently than our book does, and we're going to talk about things that all cells have, then things that some cells have, then we're going to break off into plants and animals. So all cells are going to have a plasma membrane, they're going to have cytoplasm, they're going to have ribosomes, they're going to have a cytoskeleton, and we're going to get into all of these on their own slides coming up. So the plasma membrane is that cell membrane. It is made up of 2 layers of phospholipids. It's a phospholipid bilayer. That bi means 2, right? So it's one membrane, but 2 layers of phospholipids. Remember we talked about phospholipids previously. So they have this hydrophilic head region and hydrophobic tail region, and there's 2 layers of those phospholipids there. This defines the outer boundary of the cell, and it might seem like not an important function, but it's super important. It separates the inside of the cell from the outside of the cell. It regulates cell transport, communication, adhesion, and we'll kind of talk about more of these functions in chapter 5 as well. Another thing that all cells have is some sort of fluid inside of the cell. So the cytoplasm and cytosol are that liquid portion inside of the cell. So there's liquid with some dissolved solids. It fills up the cell membrane and it helps the cell maintain its shape and cushion organelles. The ribosome then is made up of a structure called ribosomal RNA or rRNA, which we'll talk about a little bit more, and also some proteins. These ribosomes can be free floating in the cytoplasm, or they can be attached to another structure which is called the endoplasmic reticulum. We'll get into that later in this lecture. And the ribosome is important for protein synthesis. So here's the picture of the ribosome. It's made up of a small and large ribosomal subunit. And when we talk about protein synthesis, we'll talk about that a bit more. Then on this image from an electron microscope, you can see this long, like these lines here. That is the endoplasmic reticulum. And all of these little dots are the ribosomes. And some of those ribosomes are not attached to the endoplasmic reticulum, like the ones I've circled so far. And some of them are attached to the endoplasmic reticulum. So ribosomes can either be free ribosomes or bound ribosomes. And if they're bound, they are bound to the endoplasmic reticulum, which often is abbreviated ER. The cytoskeleton is a network of kind of protein filaments that are located throughout the cell. The root word cyto means cell, and then I think you probably have an idea what skeleton means. So in this case, the cell's skeleton is this network of protein fibers. It gives the cell structural support, it helps the cell have special shapes, and it also allows for intracellular movement or intracellular trafficking. So there's these protein filaments that are, in this case, this one's a microtubule, that kind of act almost like a train track, and these motor proteins help bring vesicles along different tracks in the cytoskeleton. So they can move material in these kind of little membranous sacs called vesicles from one area of the cell to another. So which organelle synthesizes proteins? That's going to be the ribosome. And what separates the inside of the cell from the outside of the cell? That's going to be the cell membrane or the plasma membrane. So that brings us to structures that some cells have. So some cells have cilia or flagella or cell walls. So cilia and flagella are these protein-based extensions, and you can see here's the flagellum. This cell has lots of cilia on it. Flagella tend to be few and longer, and with cilia there's usually lots of them and they're shorter. Eukaryotic flagella and cilia have really kind of the same underlying structure, and they were really named before we knew what they were made up of. We find them projecting from the cell surface, and they're important for movement, either to move the entire cell or organism, or to move things across the surface of the cell itself. So some cells also have a cell wall. So the cell wall is not made of lipid, and what kind of material the cell wall is made up of really depends on what kind of organism we're dealing with. So for example, fungi have cell walls made of chitin. Plants have cell walls made of cellulose. Bacteria have cell walls made of peptidoglycan. So there's a variety in the kinds of material that makes up the cell wall. So all living cells have a cell membrane, then they can have a cell wall outside of that cell membrane. So this is important because it gives the cell additional shape. Like for example, prokaryotic organisms have a relatively simple cytoskeleton, but they have these cell walls to give them their distinctive shapes. It also prevents osmotic lysis. So that one's a little trickier because maybe you don't know what osmosis is yet, but we talked about lysis, the root word lysis, before when we talked about hydrolysis. So lysis is breaking apart, and then osmosis is the movement of water across a membrane. So osmotic lysis is when a cell ruptures from too much water going into the cell. So with a cell wall, that limits the amount of water that goes into the cell. So cell walls prevent osmotic lysis. So let's review. In our pictured cells here, do they have cell walls or no cell walls? Again, pause it if you want some time to think about it. In this first picture, there are cell walls. These cells are pretty blocky looking. So this happens to be an onion root tip. In this second image, these cells are not super blocky looking. So these guys do not have cell walls. And that second picture is epithelial tissue. So like the covering and lining tissue. What cell structures move cells or move substances across cells? That would be cilia and flagella. So then the next part, we've got things that all eukaryotic cells have, then things that animal cells can have, and things that plant cells can have. I put a little scare asterisk on this all here because some eukaryotic cells are highly specialized and lose organelles as they differentiate further. So we would expect a typical eukaryotic cell to have a nucleus, endoplasmic reticulum, and Golgi body or Golgi apparatus, peroxisomes, and mitochondria. Then animal cells can have lysosomes, and plant cells can have chloroplast and central vacuoles. Plants are not the only thing that has a central vacuole. Other cells can have central vacuoles as well, but we're just going to focus on the central vacuoles that plants would have. So we've talked about the nucleus before, but we haven't really fully defined it. So the nucleus is this large, centrally located organelle. So here's a nucleus in a cheek cell. The nucleus has a membrane around it. It's a single membrane, but it's one bilayer of phospholipids. And then inside of the nucleus, there's going to be the chromosomes, which is the DNA and protein that's associated with that DNA to kind of wrap that up. So the nucleus is usually large and centrally located, and functionally it helps protect the chromosomal DNA, which in turn regulates cell function. This is kind of silly and a little bit annoying, right? But the nucleus of a cell and the nucleus of an atom, it's the same phrase, but they're both named for being relatively large for the scale of the thing we're talking about, and centrally located. The endoplasmic reticulum is often abbreviated ER. It's just a nice little shorter name than endoplasmic reticulum. The endoplasmic reticulum is a series of single membranes, so still one bilayer of phospholipids, but several membranes here stacked up to each other. It's adjacent to the nucleus, so the nucleus would be right there. And there's two different kinds of endoplasmic reticulum. There's the rough ER, which has those ribosomes on it. Remember, those ribosomes are for protein synthesis, so the rough ER has a job with protein synthesis. And then the smooth ER, it's called smooth because it does not have those ribosomes on it. And really what the smooth ER does depends on what kind of cell we're talking about. Some Sometimes the smooth ER can store things like in muscles. It's specialized for the storage of calcium. In the liver, it can be important for helping detoxify substances. Again, it really just depends on what kind of cell we're dealing with. The Golgi body or Golgi apparatus is another membranous organelle. So the Golgi body is this kind of series of membranous pancakes stacked on each other. Each of these little membranous pancakes is a single membrane with one bilayer of phospholipids. The Golgi apparatus is near the endoplasmic reticulum here. And the Golgi apparatus is going to collect and modify and package proteins from the rough ER. So right, they're coming from the rough ER, they're going in these vesicles and then going to the Golgi apparatus right there. So as far as similes or metaphors go, people often talk about the Golgi apparatus as kind of the packaging depot or shipping depot. That brings us to peroxisomes. Peroxisomes are another membranous organelle, and they contain enzymes. They can be secreted from the rough endoplasmic reticulum, and we can find them anywhere throughout the cell. They are important for lipid breakdown and synthesis and detoxification. They're named peroxisomes because they produce hydrogen peroxide. Then that hydrogen peroxide is converted to water. That brings us to the famous mighty mitochondria, right? So you've maybe heard about the mitochondria as the powerhouse of the cell before, but that doesn't necessarily really tell you what it's doing. Structurally, the mitochondria is kind of neat. It actually has two membranes around it. So it has two bilayers of phospholipids, and it contains enzymes, and it has its own DNA as well. These can be found anywhere throughout the cell, and the reason they are the powerhouse of the cell, or described as the powerhouse of the cell, is because they contain enzymes that are needed for cellular respiration, which is the process for how cells produce ATP. And ATP is kind of the energy currency of a cell. And we'll talk more about cell respiration and producing ATP when we get to Unit 2. So why can't the smooth endoplasmic reticulum produce or modify proteins? Well, kind of a big hint here is that it's the smooth endoplasmic reticulum. What does the smooth endoplasmic reticulum not have that the rough ER does have? So ribosomes, right? The rough ER has ribosomes, and ribosomes are required to produce proteins. Number 8, which organelle helps produce ATP? The mitochondria help produce ATP. So that brings us to lysosomes. In this picture, there are these kind of orange little vesicles here. So they're a vesicle, they have a single membrane, and they contain hydrolytic enzymes. So do you remember hydrolysis from chapter 3? So hydrolysis is breaking down. So this is just saying these lysosomes contain enzymes that are involved in breaking things down. They can be secreted from the Golgi apparatus. They can be found throughout the cell, as you can see in this picture here. They're all kind of throughout the cell. And they're important to clean up debris and defend against foreign material by breaking down large molecules. So this picture is kind of neat because it shows us the nucleus, the endoplasmic reticulum, and the Golgi apparatus here. And then this video is actually going to show us kind of the secretory pathway from the endoplasmic reticulum to outside of the cell. So this video is going to show that secretory path from the endoplasmic reticulum to the cell membrane, and I'm going to play it and kind of narrate through a couple things with you.
Fluorescently labeled membrane proteins start their journey to the plasma membrane after synthesis in the endoplasmic reticulum. They are first dispersed.
So in this picture, this area here is the nucleus.
Throughout the extensive membrane network of the endoplasmic reticulum, from where they move to exit sites that form in random locations in the membrane network. At each of these sites, the membrane proteins are concentrated and packaged into transport vesicles. Clusters of the transport vesicles fuse to form transport intermediates. At the next stage, transport intermediates move along microtubule tracks to the Golgi apparatus near the center of the cell.
So those microtubule tracts are part of the cytoskeleton.
The membrane proteins exit the Golgi apparatus. They move in transport vesicles that are now pulled outward on microtubules, which deliver them to the plasma membrane. Each time a Golgi-derived vesicle fuses with the plasma membrane, its content proteins disperse.
So every time one of these little vesicles fuses with the membrane, you can see the fluorescently tagged membrane proteins kind of spreading out. So there. So chloroplasts then are an organelle that can be found in plants and other photosynthetic organisms as well. So chloroplasts, like mitochondria, have two membranes around them, so two bilayers, and they contain enzymes that are important for photosynthesis. They also have their own DNA. They can be found anywhere throughout a cell that does photosynthesis. And again, they contain the enzymes that are needed for photosynthesis, which we'll talk more about photosynthesis in Unit 2. But broadly speaking, photosynthesis allows organisms to use light energy to produce carbohydrates. Plant cells also have something called central vacuoles. Central vacuoles are another membranous organelle. They have a single membrane, and in plants they're large and centrally located, and what they do depends on the type of cell in particular that we're dealing with, but typically they're important for storage of water, and they add to the plant structure as well. If you've ever had a house plant and kind of maybe neglected to water it for a bit, the plant gets a little bit limp and kind of droopy like this plant here. That's because that vacuole has lost water, so it's no longer pushing against the cell wall to support the cell. But if you water your plant, as long as it's not like too far gone, that vacuole will fill back up, and then the cell membrane will push against the cell wall, and then the whole plant will appear much more rigid or turgid. So this slide is just sort of a general overview of animal cells. So we've got the nucleus, which stores the DNA. Then here, these little yellow dots are ribosomes. So this is the rough ER. Then this other membranous structure is the smooth ER. We've got the Golgi body or Golgi apparatus, which helps package the proteins produced from the rough ER into vesicles that are sent throughout the cell. This particular vesicle is a lysosome. Lysis means breaking apart, so that's a digestive vesicle. They have an arrow to like kind of nothing, which is the cytoplasm. The cytoplasm is the cell's liquid. The cytoskeleton is the cell's supportive structure made up of all of these protein filaments here. Those protein filaments can also be used to transport vesicles besides giving the cell its kind of shape and structure. And then we've got another digestive vesicle here, which is a peroxisome. And here's the mitochondrion, which is important for ATP production. So this is kind of a generalized plant cell. In addition to the cell membrane or plasma membrane, it also has a cell wall. The plant cell has mitochondria for ATP production, chloroplast for carbohydrate production, this big central vacuole here, which stores water and helps support the cell. It also has a nucleus, rough ER, smooth ER, Golgi apparatus, cytoskeleton, cytoplasm. Let's review. Do plants have mitochondria? Yes, they do. Sometimes students get confused and think animals have mitochondria and plants have chloroplast, but remember, plants have both mitochondria and chloroplast. What are two organelles that break substances down? That's going to be lysosomes and peroxisomes. And honestly, on our test for our level, I'm not going to ask you to differentiate between the two different organelles that break things down. And then which organelles are involved in the production and modification of proteins? You may want to pause for a second so you can kind of think this one through. So where are the instructions for making proteins? So the instructions for making proteins are on the DNA, right, in genes. So genes and DNA. And in eukaryotic cells, that's going to be in the nucleus. So that's an organelle involved in the production and modification of proteins. Then we'll talk more about protein synthesis later, but kind of the short story is that DNA codes for RNA, and then that RNA goes to the organelle that's involved for protein synthesis. So that's going to be read at the ribosome. And then sometimes ribosomes are attached to what? The rough endoplasmic reticulum. And then proteins can leave the rough endoplasmic reticulum, and then those proteins are going to be carried in a vesicle to the Golgi body, and then they'll be modified and packaged there, and then they'll leave the Golgi body in a vesicle as well. So organelles involved in production and modification of proteins, we've got nucleus, ribosome, rough ER, vesicles, and Golgi body or Golgi apparatus. So kind of a neat theory in biology is the endosymbiont theory, or the theory of endosymbiosis. And I've kind of hinted at it before when we were talking about the mitochondria and chloroplast. But eukaryotic organisms are really kind of combination organisms, and they have some of their characteristics from prokaryotic organisms. And that is because a long time ago, early eukaryotic cells, they picked up like a free living cell, they engulfed it and took it in and kept it. And that first time they did that, that became what we now know as the eukaryotic mitochondria. And then plants that have chloroplast actually did it another time with a different kind of cell. They took in a free living photosynthetic cell and then eventually incorporated it into their own cells and their own bodies and kept that. So there's actually a great deal of evidence for the theory of endosymbiosis. So mitochondria and chloroplasts have many things in common with bacteria. They are enveloped by a double membrane. They have ribosomes that are similar to bacterial ribosomes, and they have DNA that is similar in structure and function to bacterial DNA. And the mitochondria and chloroplasts grow and reproduce somewhat independently inside of cells, and the way they reproduce is very similar to how bacteria reproduce. So multicellular organisms are organisms that are composed of multiple cells, right? So more than one cell. And the main benefit of multicellular organisms is that those cells can differentiate and divide the labor. So largely speaking, multicellular organisms have a larger genome, which codes for more proteins than single-celled organisms. Those additional proteins can be used for cell-to-cell communication, arranging and attaching the cells, and also specializing those cells. Multicellular organisms are, again, made up of multiple cells, but then those cells also secrete a substance called the extracellular matrix. So extra meaning like outside of the cells in this case. So this is a network of material that those cells produce, and it forms kind of a complex meshwork. This meshwork is important to stabilize cells, support cells, and organize cells. And, some tissues, the extracellular matrix is a really important and large component of those tissues, and other tissues, it's not. On this slide, I've got a picture of animal bone tissue here, and this bottom one is a cross section of a woody plant. Both of these have a lot of extracellular matrix, and the extracellular matrix is important to help these tissues be very rigid. Although broadly speaking, plants have a stronger and more rigid extracellular matrix than animals. So multicellular organisms have to have specialized junctions that hold their cells to each other and hold those cells to that extracellular matrix. So animals have more varied junctions than plants do. Animals have desmosomes, which hold cells together or attach cells to the extracellular matrix, and those cells are mechanically strong. They also have tight junctions, which prevents the material from leaking between the cells, and gap junctions that allow the direct exchange of ions between cells. In plant cells, the organization is a little bit different because they have those rigid cell walls. So plants have something called the middle lamella. Lamella literally means layer. So the middle lamella is the layer between the two cell walls in the plant. And then they also have the plasmodesmata, which connects the cytoplasm of one plant cell directly to another plant cell when that is open. So that allows material to go from one plant cell to the neighboring plant cell. So let's go ahead and label our animal cell junctions up here. So in our picture here, it's maybe a little tricky to tell. If this was like a test question, I would give you some more information besides just the picture here. This first junction does not allow material to leak between the different cells. So this first one is going to be a tight junction. The next one is structurally very strong. It's kind of like a spot weld holding the cells together. So that's going to be the desmosome. And this last one has those gap junctions that allow ions to go between the cells there. So C is going to be gap junctions. Number 13, it says label the plant cell junctions. Our options are middle lamella and plasmodesmata. So middle lamella literally means middle layer. So B is pointing to this kind of direct connection here of the cell walls. So middle lamella is B, and then plasmodesmata are these openings where the cytoplasm is connected. So the plasmodesmata is A. So we made it through the cell chapter. So we talked about the component of cell theory. So cells are the smallest unit of life. Cells come from pre-existing cells, and all living things are made up of at least one cell. We talked about cell size and scale. Generally speaking, cells are small because that means they have more surface area available to exchange materials with their environment. Microscopes in our typical undergraduate biology lab, we're going to have a light microscope. They use light to focus images. There's also electron microscopes. There's scanning electron microscopes that allow you to see surface material, and transmission electron microscopes that allow you to see inside of the cell. With prokaryotic or eukaryotic cells, what kind of cells do we have? We have eukaryotic cells. Eukaryotic cells have a nucleus, and they are larger than prokaryotic cells. Prokaryotic cells don't have a nucleus and are smaller. I already did cell surface area to volume ratio. The different cell organelles. Make sure you know the different cell organelles, be able to match them, be able to do functional type questions. So, for example, if I said the cell cilia stopped working, what would it no longer be able to do? Well, it wouldn't be able to sweep material across the surface of the cell or move the cell. What two organelles arose by endosymbiosis? That would be the chloroplast and mitochondria. And then the different cell junctions, we just went over the 3 animal junctions and 2 plant cell junctions. So that's it, and we made it through chapter 4.